Okay, so welcome to our last video lecture on translation. Translation is when the sequence of a mRNA bases is converted into an amino acid. Okay, so welcome to our last video lecture on translation. Translation is when the sequence of mRNA bases is converted into an amino acid sequence, and it involves coordinating reactions between ribosomes, mRNA, and tRNA. So let's talk about each three of these components in a little more detail. Okay, so ribosomes are the site of protein synthesis. We've talked a little bit about this, but let me show you the data that convinced scientists that this was what was taking place. And so in about the 1960s, scientists added radioactive sulfur to cells in a pulse, which incorporated into cysteine and then followed it with a chase of non-radiolabeled sulfur. And then they followed cysteine over time. And what they found was that the sulfur gradually accumulated with ribosomes, and then that association then decreased over time. So basically, ribosome is where protein synthesis was taking place, but then once the protein was made and it was a free polypeptide, it no longer associated with the ribosome. So in bacteria, this process of transcription and translation that's happening because of ribosomes can occur simultaneously, and that's because there's no nucleus. And so the mRNA doesn't have to be transported out of the nucleus. And so here's an electron micrograph showing DNA right here, if you follow along. And then the mRNA, you can barely see it, but it's where all these dots are. And these dots are ribosomes bound to the mRNA. And so multiple ribosomes attach to a single mRNA to form these polyribosomes. And many copies of a protein are produced from one mRNA. And this is depicted down here below, right? So you have RNA polymerase making the mRNA in bacteria, then a ribosome is binding to that and then synthesizing the protein. And as this mRNA gets longer, more ribosomes can bind, right? So that you have multiple proteins being made off of this single mRNA. And again, this is happening in bacteria, but in eukaryotes, the mature mRNA, right, they've already been processed, as we talked about last lecture. They're transported in the cytoplasm where translation by ribosomes and formation of polyribosomes can occur. Okay, so you should be comfortable kind of figuring out what you're looking at in this picture. So we're talking about the process in which mRNA is converted into amino acids. And one early question was, how does mRNA specify for an amino acid? And there were two existing hypotheses. One is that amino acids interacted directly with mRNA and codons. And the second hypothesis was proposed by Crick, and he proposed that an adapter molecule was holding on to the amino acid and was holding it in place, and the adapter molecule was what interacted with the codon. And this might have made sense because he thought that this adapter molecule might be able to form hydrogen bonds with the mRNA. And so scientists discovered that these transfer RNAs or tRNAs were delivering amino acids to the polypeptides. So I'll present some of the data kind of supporting that the transfer RNA served this role. So the question was, what happens to amino acids that were attached to transfer RNAs? So we had worked out that amino acids attach to these RNA structures. And so what they did was they radio-labeled leucine, so you could follow leucine, and they allowed those to attach to tRNAs. And then they put it into a solution of mRNA and ribosomes. So when a tRNA is attached to a amino acid, we call them aminoacyl tRNAs. And we put it in this test tube, and we call this test tube an in vitro stands for in test tube, solution. And then we followed the fate of radioactive amino acids over time. And the hypothesis was that the radioactive amino acids will be found in polypeptides so that they would begin to polymerize. So this shows, so I'm going to show you, follow these gray dots first that are all highlighted in yellow. So initially the radioactive amino acids started attached to the tRNAs. And then gradually they were no longer attached to the tRNAs. Instead, the radioactive amino acids were gradually incorporated into polypeptides. So going back to this previous experiment or this previous hypothesis, it appeared that these tRNAs were this intermediate. So how are they doing this? So let's take a closer look at tRNAs. tRNAs, as I mentioned, stands for transfer RNA, and they are the adapter protein used in translation. An aminoacyl tRNA is the one shown here, is the one where tRNA is linked to its amino acid. tRNAs are relatively short. They're 75 to 95 nucleotides long, and they form this important secondary structure by folding into stem. This is the part where they form hydrogen bonds between each other. So stem, loop, stem structures. At the end, so it has complement, or it has polarity. So you have a 5' end that wraps around to our 3' end. And at the 3' end is the sequence CCA. And this conserved sequence is the site at the 3' end where the amino acid attaches at this binding site. And here at the bottom, right, is where on the opposite end is where the anticodon exists. So the anticodon is the antiparallel complementary sequence to the codon. So it can base pair with the mRNA codon in this antiparallel manner, and then that's what brings in the matching amino acids. So a pretty cool process. How are amino acids added to tRNAs? This actually takes an enzyme called an aminoacyl tRNA synthetase. Okay, so that's a long enzyme name. But the enzyme is shown here in green. And what happens is that a tRNA, right, that 3' end of the tRNA sticks into the active site, as does an amino acid. And this enzyme, we say that it charges the tRNA. It causes these two to form a bond at the CAA, right? It forms a bond. And it catalyzes that attachment of the 20 amino acids. Each different, there are 20 different amino acids, and each has its own enzyme, so its own aminoacyl tRNA synthetase. For each amino acid, there is more than one tRNA. So there's lots of different tRNAs, right, because they have the anticodon. So there's lots of these enzymes to attach the enzyme, or to attach the amino acid to the tRNA, and this process does require ATP. So it takes energy to make proteins. There are 61 different codons, but there's actually only about 40 tRNAs in most cells. And so this is kind of problematic because we seem to be missing some tRNAs. And so Crick proposed this idea of wobble pairing as a solution. So remember that this third position in the codon tends to be the one that changes. And so Crick proposed that the anticodon's third position can form a non-standard base pair, and so one tRNA is able to read more than one codon. So here's an example of that here. Here we have a codon that's CUC, and the anticodon is GAG. This is a normal base pair. But in wobble base pairing, we can have a non-standard base pair between a G and a U occur, because they'll kind of maybe separate a little bit, but this codon will still bind to this particular anticodon. And this is known as wobble base pairing and solves this problem of not having enough tRNAs for every codon. Okay, so we've been talking about tRNAs. Next, let's talk about the other key player, the ribosome. So ribosomes, as we've mentioned a couple times in the course, contain many proteins as well as ribosomal RNA or rRNA. And the ribosomes can be separated into two subunits. We have the small subunit, and during translation, this is the subunit that holds the mRNA in place. And then we have the large subunit up here, and that is where the peptide bonds form. Now this is a very busy diagram here. We're going to go over all the parts. But again, we just have the small subunit holding the mRNA and the large subunit, which is where the peptide bonds will form. During translation, 3 tRNAs are going to line up within the ribosome. And these tRNAs fit into 3 different sites within the ribosome. And they will form antiparallel complementary bonds with the corresponding mRNA codons. So there's, as I mentioned, there are 3 sites. The first site I'm going to talk about is the A site. And the A site is the acceptor site or amino acyl, site. And this is where the tRNA comes in to the ribosome and it carries an amino acid. The P site or peptidyl site holds the growing polypeptide chain. And then the E site is the exit site. So tRNAs without amino acids, they're no longer charged, exit the ribosome. So we have acceptor and essentially peptidyl or peptide, and E for exit. So those are the 3 important sites to remember when talking about what's going on inside the ribosome. Okay, so we're going to talk about the steps in which polypeptides or these codons are converted into polypeptides. And this occurs in 3 steps: initiation, elongation, and termination. So basically the same exact steps as transcription. Different process though. So the first thing that happens during initiation is we have our mRNA shown here. And here in our mRNA is our start codon, AUG, okay? And about 6 bases upstream from the start codon is the ribosome binding site. And in bacteria, this ribosome binding site is also known as the Shine-Dalgarno sequence. You can look up a lot of really funny memes if you search Shine-Dalgarno sequence. And so this ribosome binding site attaches to the ribosome, and it does this with the help of these initiation factors shown here in green. So these are proteins that associate with a small subunit. And this brings in that mRNA. So now we have the ribosome binding site, attaching to the ribosome. And because of these initiation factors. And then the next thing that will happen is that the tRNA carrying the start, that recognizes the start codon. So here we have our start codon AUG. So the anticodon is UAC. And that's in the tRNA that's attached to this special start codon. In bacteria, it's a modified methionine called FMET. And then once that attaches, the large subunit of the ribosome binds. and this completes ribosome assembly. And so you'll notice that this start codon is loaded in, when this large ribosome subunit binds, it's in the P site. So it's right there, right? It's the one holding the amino acid. Okay, so the next step is elongation. So when we're getting translation occurring, the amino acids are always going to be added to the C terminus of the polypeptide chain. So here's the N terminus, here's our carboxyl group. This is another N terminus of this amino acid. We're going to get a peptide bond right there. So what happens during elongation is that the next tRNA comes in at the acceptor site, the A site. So it's going to come in at the A site, and amino acids in the P and A sites, right, will be in the active site of the ribosome. And this is where a peptide bond will form. So we'll have a condensation reaction occurring between this F-met and this glutamate. Our RNA in this active site is actually what catalyzes this peptide bond. And so a ribosome is also known as a ribozyme, and that's a term that refers to RNA that's catalytically active. So now, right, you'll notice that there's no longer this tRNA in the P site. It's no longer holding on to the amino acid. And so we have this transfer of this amino acid to the tRNA that's in the acceptor site. And so what happens very quickly is translocation. So what happens is that uncharged tRNA is going to slide over into the exit site, and then the tRNA that was, right, in the A site is going to slide over into the P site. And when this happens, it's also going to slide the mRNA with it. And this is going to reveal essentially a new codon that can be recognized with an anticodon. And this is helped along by elongation factors. So translocation accomplishes two things, and I'm just going to summarize these. Three things. The uncharged tRNA from the P site, that was here, moves to the E site, right there, and it's eventually ejected from the ribosome. The tRNA attached to the growing polypeptide moves from the A site into the P site, and this opens the A site again for a new amino acyl tRNA to come in. So this process is going to be repeated over and over again to grow the polypeptide chain. So here we have a new tRNA coming in with its anticodon binding to the codon. We have the formation of a peptide bond. So now the peptide is transferred briefly to the A site, or to the tRNA in the A site. That one goes ahead and slides over to the P site, and the one that was in the E site is ejected. So this will move on until we encounter a stop codon, such as UAG. And this is where translation is going to stop. So there is a protein called release factor, and this is going to come into the A site when the mRNA has a stop codon. And this is a very cool protein because it resembles a tRNA in size and shape. And it hydrolyzes the bond, so there's a bond right here, between the leucine, right, and the tRNA. And the release factor is going to go ahead and hydrolyze that bond, linking the tRNA and the peptide together. And so it's going to release the polypeptide. So the newly synthesized polypeptide, the tRNAs, and the ribosomal subunits can all separate from each other. So that's the end of translation. So most proteins do go through a few more steps before they're functional, and this process is called post-translational modification. This can include things like the addition of sugars, and lipids, as well as the addition of phosphate groups by enzymes that can alter the protein's activity. And these steps can take place in the cytoplasm, but some proteins need to be moved to organelles like the Golgi to be modified. So just to review kind of what's going on in the ribosome, we have an amino acyl tRNA entering the A site, and it's going to remain there if the codon-anticodon matches. The peptide bond then forms between amino acids in the A site tRNA and the polypeptide on the P site tRNA. And then the ribosome moves down the mRNA one codon at a time, and all 3 tRNAs move down one position. The tRNA in the E site exits, and that creates a new site in the A site for another tRNA to bind. So that's it for this video. There's only one video today, so I look forward to seeing everyone in class. Thanks, bye.